Next, we're going to ask Dale Griffin to come and talk. He is a environmental public health microbiologist. He's at the U.S. Geological Survey, and he has particular interest and expertise in vector-borne diseases.
Okay, Martin, how much time do I have? 12? Okay. Okay. So I've got 28 slides, so I'm going to cruise through some of these fast. My name's Dale Griffin. I'm an environmental public health microbiologist with the United States Geological Survey. And myself, along with some colleagues in the EPA and a couple other scientists at the USGS, have been working at looking at the distribution of pathogens in the continental United States. What are the constraints and so forth. So what I'm going to do is give you a real quick introduction to my journey through the valley of big data. So, you know, one of the big needs in environmental microbiology is understanding, when looking at disease and outbreaks, is understanding what's the natural background distribution of these various pathogens. I mean, there's nobody in this room, and I know the data doesn't exist anywhere, that anybody can tell me what the natural background distribution in the lower 48 in North America is for any given pathogen. Nobody can. I mean, you might be able to tell me that Ebola's not here, but you can't tell me something that's here. What are the constraints? What are the geological limits, climate limits, and so forth of these organisms? And even if you had that data, what you really need to know to determine risks to any given population, whether it's livestock or humans and wildlife or so forth, is how geochemistry and climate controls these organisms. So that's what we're really focused on. Really, I'm going to give you a two-part story. One is how we got started and generating some of this data and what we're doing now, and as far as developing a GIS-based model, an interactive model to serve as a guide, to kind of guide you along investigative paths. So it started with the USGS Geochemical Landscape Project, which ran from about 2004 to 2010. It was run out of Denver, Colorado. Dave Smith was one of the lead authors on this, and the goal was to determine the geochemistry of souls across North America using about 5,000 sample sites. This is a distribution of sample sites across the country, and this is some of the elements we are looking at. So 44 different elements, and also mineralogy. So you're looking at determining quartz and stuff such as total carbon and feldspar and so forth. But my interest in this was I've spent a lot of time in the laboratory working on a lot of different media types. I know the limits of culture-based assays and so forth, and anybody who's taken a course in microbiology knows you use this media type for this organism and that media type for that organism. And if you look over into the left, the TSA, that's a common nutrient, very high nutrient agar. And the bacteria are doing really good on it with some fungi, but if you go to a different media type that's focused more towards fungi, like mycophyll, you can see the fungi are growing, they're very healthy, growing very fast on there. So really the nutrients control distribution, and my kind of perspective as a microbiologist, but if you take a bird's eye view of the United States and we're looking at the distribution of phosphorus in our soils up there, is you can view that as a culture dish and view it as a very, somebody in the laboratory didn't mix the media very well, because you can see the heat map shows that there are variations across the country on the concentrations of phosphorus in our soils, as you would think. And if you go through the list of all those 44 elements that we are looking at, every heat map is different. So, and I guarantee you that this greatly influences the distribution of the organisms that live in our soils, such as anthrax. And that was kind of a focus of ours early on, following 9/11. And my job was really to, and this is historical anthrax data, was to go through those 5,000 samples and use the polymerase chain reaction and look for anthrax and bacillus in general in our samples. So this was a pilot study early on. You can see a transect that went from Manitoba, Canada, all the way down through the central part, down to about El Paso. And these, the squares are where we found bacillus and anthrax, and the diamonds are where we found just Bacillus species and not anthrax. And the little circles, we didn't see either one. But you can see a distribution up in Manitoba and down in Minnesota where outbreaks occur annually and so forth. So that was the pilot study, and we actually went into some soils after Katrina, and 5 of the samples were positive in the muck that was in Katrina, which was a combination of seawater and wastewater and so forth. And it was interesting because the one thing they say about anthrax is the spores, once they're there, they're supposed to be, they're supposed to stay there forever. And what's dogged a lot of people who study this organism is you can go back, you can have a bison that dies in Montana, and you go back and he rots on the ground. He's got millions of vegetative cells that sporulate and are deposited in the soil. You go back 6 months to a year later and you can't find spores. So where are they? What's happening? And I can tell you that we really don't understand. So my perspective was, well, let's take not only a point data of the samples we were collecting. Well, that's one avenue we can look at the distribution if your assay is sensitive enough. But what we have that's readily available is outbreak data. And so the red counties, the counties in red are where we have had outbreaks of anthrax since 2000. Okay? And so every year, I can guarantee there's outbreaks of anthrax in this region of Texas and up in this tri-state region of Minnesota, North Dakota, and South Dakota. And you get sporadic outbreaks out in Klamath County. They have one every once in a while, and you see some in different counties around. But these are the hot spots, southern Texas and up here. And what the Geochemical Landscape Project, each one of these counties has multiple sites for geochemical data. So my take was, well, let's go into each one of these states, take the red counties, compile the geochemical data for the positive, and then contrast within the same each state and counties that have never reported during this period outbreaks, and see if there's any geochemical differences, and just do the stats for this, and then the positive versus negative counties. And what we saw was we identified a number of elements that were statistically significantly higher in positive counties than in negative counties. And you see the red counties and blue counties are illustrated on here. Calcium, I think Louis Pasteur even noted that calcium influenced the prevalence of anthrax in the 19th century. So that was widely known. It was nice to see that the stats showed that. Phosphorus is another thing that's been known for quite some time to influence anthracis. Manganese, strontium is a spore element used in its spore, makes it more heat resistant. That was interesting to see. And zinc is critical for the lethal factor, which is one of the toxins in anthracis. So it was also interesting that we saw some elements that had an inverse relationship. So the higher the concentrations, the fewer you wouldn't have outbreaks of data. And that was barium, which is closely, if you look on the periodic table right here, closely related to calcium. So it may mimic that element. So anthraxasis may pull it up, and it may block it from prospering. And then you go down here to strontium, which is one of the positives that we saw, and rubidium is right next to it, which is one of the negatives. So that was very interesting. And this is just a total carbon heat map. This is pretty interesting. This hot area down here in southern Texas is very high in total carbon, and this region up here in that tri-state area is very high. And so this model we're developing has a swipe capability, so you can go in and you can outline your counties, and you can take one of your elements, and you can swipe over it and go back and so forth to look for patterns. So you can cycle through all these different elements to try to develop a list for what you may want to look at in a lab-based study. And then you may have some interest in soils and pHs and so forth. So this is 2 different data sets. One's Sergo and one's StatGo. The Sergo has some gaps out here in the Midwest, so if you were looking for the valley fever agent, coccidioides amylitis, you see some gaps in the data. So you might want to go with StatsGo. The resolution's not as good. It's on a scale of 1 to 250,000 versus 1 to in the 50,000 range. And if you cycle over and swipe over, you see, well, StatsGo's got good coverage in that area, so I might want to use that data set. So this is a way to use the model to look for different data sets. And we heard this morning talk of zoonotic transmission, we all know how important that is. And so these are, this is a census of our beef cattle So there may be some, anthrax is certainly one organism of interest, foot and mouth disease and so forth. And we have this for bison, and there's Fish and Wildlife has developed some data sets for many different organisms. We have one for deer. The bison one wasn't complete. They had a list for all the domesticated bison, but they didn't have free ranging. The populations. They didn't have anything for the Yellowstone area, and Stephen Douglas, who put it together, was like, oh, Dale, something's wrong here, you know. So we found out that their data set didn't include free range. They were just captive populations. So that's an option. And this is precipitation data. You can see 1960s versus 2000s. You can see a change in the amount of rain in the areas of the country that are getting more rain. It might affect the distribution of various pathogens. And this is the PCR data across the country. And it's interesting, if you go up in the Montana area, that there's not many hits. But we have hits throughout soil. So bacillus species in general are very prevalent in our soils, and about 0.03% of these were positive for anthrax. And I presented this data, the anthrax data at the CDC up Atlanta at a meeting, and one of their groups, their Naegleria groups, this is a nasty little amoeba that's known as the brain-eating amoeba. It's very lethal. I think since 1962, there's been 138 cases, only 3 survivors. So you get the infection. It's a lot of times it's kids that are playing in shallow, warm waters. And I said, well, you know, can you give me the historical outbreak data, which is what you see here. And the browns, which are down here in Florida, and the dark browns and up here in Virginia are from the 60s and 70s. And then we started getting all these hits in Texas that popped up later, and the dark greens are the latest that went all the way up into Minnesota. So I just want to show you some stuff on this. It's pretty interesting. So you can take this model and highlight all these sites and ring them with the watersheds. If you're interested in the watershed, you can add your data to include your rivers and lakes. And you can zoom in on these sites, click on individual points, look at the metadata associated with each data point. And a lot of these in Texas are associated with reservoirs that they have out there. And then you can go into land use and highlight each one of those water basins and look at different land use trends and so forth. And this is really interesting. Copper. We know copper has a detrimental effect on certain microorganisms, and where copper is typically low, what you're seeing here is where you find this pathogen. You have a couple of anomalies out here in California and Arizona. If you cycle down and go to look at zinc, which is another that's pretty interesting, a trend down here through Florida and along the East Coast where we saw the earlier outbreaks. You go out into Texas, and most of the outbreaks occur in regions outside the hot spots that are in Texas. And then you go out here into Arizona and Southern California, and those outbreak areas are on the fringe of hot spots. They're not in it. So that's something we think may be important with this pathogen, is the concentration of zinc. And if you look at another thing in this California-Arizona region, you look at the change in climate over time. So this is the difference in temperature from the 1960s to current. The darker the color, the greater the temperature change. And the Minnesota outbreak and the California and Arizona outbreaks, some of the later outbreaks have occurred during this period, so there's some evidence of range expansion. Okay, and this you can see in the 1960s when you had that high one up in Virginia. In the 60s it was warmer. Remember Leonard Nimoy telling us that we had an ice age coming in the 70s? Well, the temperature had dropped, but then there's been a steady creep since then. These are the average latitudinal positions of the outbreaks for each decade. So there's been a steady creep north since that high in the 60s, so that's pretty interesting. So this is a web page we'll have up soon that will describe the work we're doing, and it'll have this model will be available online pretty soon for public, academic, and other research use. And that's my talk. You know, we've seen that if you can take some of these data sets and create layers and for overlays on various pathogens, it's been very productive, and I think it's just a great decision support tool. So I hope you enjoyed it, and thank you for the opportunity. What's that? Okay. Any questions that I can answer? Yes.
Thank you very much. Very interesting presentation. I'm just wondering, when you start combining the number of pathogens and the number of elements, you end up with a very large number of potential combination. And when you look for associations, well, in statistical terms, it's a bit of a fishing expedition, really, and you're bound to find something. So how do you combine looking at all this different combination with biological plausibility and some kind of biologically plausible mechanisms to identify what is real associations versus just, chance findings.
Yeah, I think it's best to use as a decision support tool, kind of a guide. And you're right. It's like the face on Mars. It looks like a face. It's not really a face. And you'll get that with that much data, and you throw out there, you'll see some relationships that just aren't real. And so I think that's the value is to guide you to a path where you're not looking at everything, you're looking at a fewer group, and can eliminate those through a series of maybe lab-based experiments or so forth. I remember presenting this data with the strontium, and there was a senior researcher in the audience who met me at lunch, and he was like, Dale, of all the years I worked with anthrax, it never occurred to me to look at strontium, but that's a critical element in the spore when it forms spores, so. Surely, there's shortcomings, yeah, but I think there's great value in it. Anybody else?
Fantastic. Thank you, Dale.
Thank you.